Hey Jordan.
Hello.
Hey, how are you? Good to see you. I
Good.
don't I don't think we've ever met each other before, have we?
No, I don't think so.
So I'm Mark Billinghurst. I'm the director of the lab here. Normally Aladin would be running these talks, but he's on a flight right now to Canada. So he's actually mid-flight, so he's got a good excuse
Oh,
for
cool.
not being here. Yeah. Um,
Where's he going in Canada?
Toronto.
Oh, cool. Okay. I'm from Nova Scotia originally, so I
Oh
was
wow.
just
So
curious. Yeah.
it's pretty close. Yeah,
Yeah.
I've never been to that part of the country. Most of my time in Canada has been, well, I've been to Toronto, but mostly on West Coast and Vancouver. But I've heard
Right.
Nova Scotia is very beautiful. So, so you're in Northeastern, so that's in Boston, isn't it?
Yep. Yep.
Great. That's another one of my favorite cities. It's a great place to be. And I'm not familiar with your institute, the RU Institute. Is that part of a neuroscience institute, or is that its own
It,
research institute together, or is it part of a department? Or...
so it's kind of it's a sort of project that Northeastern's trying to start up where I've technically got my postdoc right now, where they're trying to combine machine learning and sort of experiential AI is what they're calling it with like they're trying to sort of set it up with business partnerships and things like this as well. The
Hmm.
most likely I'm probably my postdoc's just about up with them anyway, so I'm probably actually going to be working at MGH with the Martinez Center soon. So
Oh,
which
the
will be
hospital.
perfect for the
Yeah.
yeah, which is going to be perfect for the sort of imaging work that I'd want to be doing anyway. So
Oh, okay, that's interesting. And how do you pronounce your last name? Is it Theriault?
so Theriault.
Theriault. Okay, sorry about that.
Yeah.
That's
No
it
worries.
looks like it's a little bit French. I'm not sure. That's
Yeah,
okay.
it's long since anglicized. It was Acadian in Nova Scotia, and then
Oh,
basically we
right.
lost all of the French in my family for a while now. But
So it's really great to have you come and give a presentation. Your presentation is going to be a little bit different from some of the others we've had. So I don't know how much you know about our research lab, probably not a whole lot, but we are in the Auckland Bioengineering Institute, which is the largest institute in New Zealand for bioengineering. But
okay,
our research is more in the computer interface area, although we have a strong interest
interesting.
in brain activity, and we're doing research
Right,
now on looking at EEG with augmented reality and virtual reality, both from the perspective of measuring people's cognitive load and while they're performing tasks, but we're also interested in shared EEG activity, in particular using two people doing the same task together and measuring brain synchronization.
right. It's
So
hyperscanning, right? I think
yeah,
I saw that you
hyperscanning.
guys were.
Yeah,
Yeah,
yeah. So the hyperscanning, which is I guess it's been around in the psychology literature for about 15 years or so. It was really expensive until recently to get EEG equipment or FNES equipment, and it was and because of that, it was hard to have people do studies at two people at the same time using
right,
equipment. But now the cost has dropped. And so the last 10 or 15 years, people have been starting to study this now, and it's become more popular. But there's almost been no research at all been done in the AR and VR space. And so we're
right,
just looking at that. But both from a perspective of looking at how we could use, so with obviously in the real world, it's quite difficult to modify people's appearance and see if that
right,
affects brain activity. But in VR, you can do that. You can have different types of virtual characters, and you could change if you can see that people's mouths or eyes or whatever, and you can see
right.
if that's having some impact. So we're trying to explore that, but it's still early days. Though we've got a couple of papers published, but we've got a lot more to do. So your work's probably, and then we're doing some work also on the affect and emotion side. In fact, Cornell, who's just
Right.
joined us, his research is on measuring using context in VR to measure emotional response to VR experiences. So,
Hi,
so,
Jordan.
so
Right.
yes. So some of what you're talking about seems to touch to that space as well. We haven't been approaching it from a metabolic perspective, but although in the overall institute here, there's a lot of interest in that. In fact, I just came from a talk. They've got a very large project in the institute about trying to map all the body's metabolism and connect that up to
Interesting.
physiological structures and the like. So it's a very big project. A long time ago, they did a very nice project on building mathematical models of the heart and lungs, and now they're trying to extend that to the whole body, basically, which is going to be a huge effort. Yeah. So there are some people here who do a little bit of fMRI and some neuroimaging, a lot of CT and you know, body imaging as well. So, yeah.
No, that's really, really cool. I wanted to ask as well how you guys had found me in my research. I'm wondering if it was through the sense the should stuff or the.
I think it was. I'm not. Canal, I think it was it Tamil who recommended Jordan, or we've got another student who's
I think Reza might have.
Oh, that's right. Yes. So we have a student called Ali Reza who's looking at machine learning models for emotion, combining EEG and FNES. So we've been asking our students on a regular basis who would be great people to have come and talk. And now with COVID, you know, people are very comfortable at giving remote talks. So it's a really great opportunity for us to kind of invite the best in the world to come and hear from them. And so that's
and I think you work with Lisa Feldman-Barrett, who has been inspiring. So her book How Emotions Are Made was the first detailed scientific book that I've read and learned a lot. So she is an inspiration to us and would be. And like, so I'm considering past experiences as contextual information, and from that I got inspired, like how past experiences influences the current emotional states. So it's pretty fascinating having.
No, I loved her book. Her book was what sort of drew me to her in the first place as well, because I think I joined the lab right at the time where she'd had the book come out, and there was a scan article on the theory of constructed emotion, and I was sort of feeling the disillusioned with social psychology at the time, I think, and then sort of saw her work, and it was like a big breath of fresh air, and I really, really loved working with her.
Yeah, would love to hear more stories about how she came up with that solution. Like, because theory of constructed emotion is really amazing, and it's different and very practical.
Yeah.
So
no, absolutely.
Especially with the affective computing direction, which has been going, like with the Paul Ekman's theory of that 6 basic emotions and facial expressions. So I find that really interesting how she came up with the theory of constructed emotion, and that's my path that I'm trying to follow and try to use that in virtual reality.
Right. No, that's really, I mean, there's been a lot of work. I think we've been really interested in doing virtual reality work as well too, although we have some colleagues at Northeastern who do it, but it's I think we had a big project that we'd wanted to work on a long time ago, thinking about how to sort of tailor a personal experience for some, like a lot of the work that we've been doing has been collecting real world like physiological sampling. So we'll sort of suit people up with physiology gear, send them out on their day. And then if there's a sudden change in physiological signal that's not due to motion, then people are basically tagged to report on whether they're feeling an emotion at the time, what's going on in their context. And we've been interested in doing something like taking that sort of real world experience sampling and then trying to like recreate real world situations in a virtual environments that you can start to experimentally tailor like bits and pieces of them or something like that too. But
yeah. So in your the experiment that you just mentioned, when you say context, so what kind of information do you collect, like location and activity or anything else specifically that you are looking into?
Yeah, I think for the stuff we'd want to do that would be virtual reality, which we haven't done at all yet, we need a lot more context than we collect right now. The context that we collect right now is mostly the physiological context and then what people report on. It's more of a study for looking at like emotion granularity. So whether there's it's basically like the strongest test that you could look for of sort of the Ekman basic emotion ideas, that there's like a real one-to-one physiological signal for like a specific emotion. And it's turning up that, yeah, there's definitely not. there's like a lot of different ways that the physiology can go that people classify, and you'll have big differences between people as well in terms of like, some people will classify every single emotion experience as something completely different. Some people will just classify it as like positive, negative, feeling good, feeling bad. But
so I'm more looking into like a valence and arousal dimension, not just like the discrete ones, because I don't know. So there was a sentence in the book by Professor Lisa Feldman Barrett that like the like it's priming if you show them the discrete words. If you ask people like, are you feeling happy, sad, or so, then they just feel, okay, these are the options that I have to select, not much. So yeah.
So Jordan, just want to check, can you let me just make you co-host so you can share you just make sure that's all working. We got a few more people just coming in, so just make you co-host. So can you share your screen just to make sure it's working okay? And we'll wait a few minutes before you get started because we normally have more on the call. So we've probably got a couple. So yeah. Oh yeah, that's perfect. That's working great.
Is this working?
Yeah, I can see the whole. Yes, that's great. That's looking great. Excellent. So just wait a couple more minutes. We've got, my lab is split between Australia and New Zealand, and so it's a little bit earlier in Australia. So it's 10:30 in the morning in Australia. So some people may be coming in, and then in New Zealand it's 1 o'clock here. So I guess it must be late in the
9
evening.
o'clock.
Yeah,
9 o'clock. Yeah.
Thanks for staying up so late. Appreciate that.
No. I was scrambling to get the slides together all of today. So you're catching me at the end of a long day here.
Oh, yeah. Well, you can sleep after this. It'll be great.
Yeah.
Yeah. So let me just check around. I think most everybody in the lab who's physically here is on the call. Wasn't sure if not joined. And then we'll see, we might get a couple more from Australia. We also have an intern program. We invite our interns, but we normally get between 10 and 20 people on the call, and we also record the talks as well, if that's okay.
Okay. Oh yeah, of course.
We put them on our website after the fact. Some of our students and others are different time zones. So
for sure.
Yeah. Okay, well, maybe we'll get started. So I'll just give you a brief introduction, then you can talk. So we're really grateful to have Jordan presenting with us today. And Jordan's a postdoc at Northeastern University. He's doing a lot of interesting research in neuroimaging and in models of affect and emotion. So I think he'll talk about today a little bit about the metabolism, or I guess I can read from your abstract here. Activation-based aerobic glycolysis is an unusual and energy and inefficient form of glucose metabolism that usually occurs in tandem with stimulus-elicited bold activity. So he's going to talk about how this enables us to interrogate brain activity and how this also relates back to computational models of effect and motivation. So I'll turn the time over to you, Jordan, and we'll basically have a bit of a talk for 45 minutes or so, or an hour, and then we'll have some time for Q&A at the end, as much as people want to. So thanks very much for your time, and I look forward to hearing your presentation.
All right, perfect. Yeah, so it's going to be, forewarning too, that it's going to be pretty much a theoretical talk. A lot of the stuff that I've been doing has been sort of theory-based work, but we're trying to, which I know is a little unusual for social or for psychology talks a lot of the time, but hopefully bear with me and I'll try to make it make sense here. Cool. 1 sec. I'm going to try to, sorry, 1 sec. Before I start up here, I'm just going to hide the meeting controls here so that I can work on this. Okay, is this still showing?
No, I can't see anything. It's full screen to me.
Is the screen sharing though?
Yes, screen sharing is fine. The meeting controls is gone, so it's looking great. Thank you.
Perfect. Okay. So I just wanted to start out with a couple of quotes here that I think are really important for thinking about how to scaffold a theoretical research program that I'm going to try to walk through here. So the first one is, if people are familiar with Ash, a famous social psychologist, he talked about how the psychologist whose concern is with individuals and society takes as his task, or her task, the study of the structure of forces within individuals that make social orders and regularities possible. Oops. Then at the same time, there's a quote from Kenneth Gergen, who's one of the original sort of thinkers of constructivism in psychology, who talks about, due to the relative stability and simplicity of the biological system, as contrasted with the vicissitudes of social history, the biological system would seem to furnish an enduring backdrop for the shadow show of mores, styles, and custom. And so the reason I wanted to start with these is the first quote I like because it's a nice inversion of the idea that psychology is reducible to biology. Ash had actually also talked about the idea that psychology could be considered a physics of the social sciences, and that if we could understand fundamental forces in individuals, we might try to get a grasp on how they work in interactions or in groups. But then at the same time, the second quote I wanted to start with, because it clarifies that to get some traction on individual psychology, we're going to need to incorporate a biological understanding, even if the psychology itself can't actually be reduced to it. So there's social features that are going to vary across people, places, cultures, and historical moments, but these are implemented through our biology. And so biological understanding can help make that research actually tractable. And another thing, just to start out, because my background's a bit of an odd mix talking about social psychology and metabolism, so it maybe helps to just give an introduction of where I've come from and how I got to here. I'd worked on social and moral psychology with Leanne Young at Boston College. And then despite that initial interest in that, I've sort of transitioned down the road in Boston here to start looking at affective neuroscience. Oops. To look at affective neuroscience and emotion with Lisa Feldman Barrett. And when I got there, she'd really encouraged me to take a biological perspective, dig into brain metabolism. I was skeptical at first, but it's really paid off, I think, because we've dug deep into thinking about these biological systems that make the social systems tractable. Cool, and so the talk itself here, I'm gonna combine some thoughts from two different papers. One of them we have up as a preprint that's thinking about the brain metabolism side. The other is metabolically based, but it's more about the social psychology side of it. And again, this is my first time actually trying to combine both of these into one talk. And so if there's gaps or if you have any questions, you can feel free to stop me if it's really pressing and I can try to clear it up. Cool. So I'm going to try to organize the talk here around 3 questions. The last 2, I think you're going to find intrinsically interesting. And then the first one I'm going to try to convince you is actually fundamental to answering the other 2. So interpreting bold activation, modeling social motivation in metabolic terms are very flashy, but we kind of have to start with the fundamentals of understanding brain glucose metabolism, which is going to make the rest of this possible. Again, sorry, I'm going to try to close the meeting. Floating meeting controls here. Okay. So again, another quote, why focus on brain metabolism? So this quote from Hermann Ponzer talking about at its biological core, life is a game of turning energy into offspring. And what I mean to take from this is that from an evolutionary perspective, energetic efficiency is actually really, really critical. Energy wasted is energy that's not available for mating, caring for offspring, seeking nutrients, or escaping predators. Metabolic costs are going to be a factor across your entire lifespan, unlike reproduction as well. This is a sort of, both are going to be essential to evolution. And you can consider metabolic efficiency as a slow, steady, and constant pressure shaping evolutionary design. But reproduction is something that's going to be done when you have energy to spare. And I just try to put this up to dispel when people are thinking more of in an evolutionary perspective, people tend to be very fixated on reproduction and not think again about the fundamental factors that shape the way that the brain's designed. Cool. So again, the brain itself is actually a major metabolic investment. And so just to begin, if we were to think about rat brain, chimpanzee brain, and human brain, In the rat, the brain metabolic rate is about 5% of resting oxygen consumption. In the chimpanzee, it's about 9%. And in the humans, it's about 20% of resting oxygen consumption, which is most of the metabolic consumption, or a pretty good index of metabolic consumption is taken up by the human brain. Among that, neuronal signaling is pretty much the major cost of that baseline metabolic investment. So if you were to total it up based on current estimates, neuronal signaling would account for about 13% of the human whole body metabolic budget, which is just quite massive. And again, the majority of that is going to the sodium-potassium pump and essentially repolarizing after action potentials, after synaptic transmission. Very little of it is going to biosynthesis on a moment-to-moment basis. Across the whole life, the major investment tends to be this neuronal signaling. And again, I got so many quotes at the start here, but I can't really help it. Going back to Darwin here, there's a nice point that he makes, which is that natural selection is continually trying to economize in every part of organization. It'll profit the individual not to have its nutriment wasted on building up a useless structure. So keeping that in mind, it might come as a surprise that the bold activation that we're actually all used to measuring is an index of metabolic inefficiency. So for people who forget, the way that the bold activity works is if we were to show someone a stimulus, let's say a flashing checkerboard here, there's going to be an increase in blood flow. There's going to be a slightly lesser increase in oxygen use, which produces excess oxygen, which is what produces the bold response itself. So again, for people familiar, positive bold signal is when blood flow exceeds oxygen use and a region's flooded with oxygenated blood. And we get the classic bold response that we see here in response to stimulation. Again, the way that people talk about it is they'd say blood flow decouples from oxygen use, and this produces the positive bold signal. What's less often talked about, but what's actually present if we go back to this Fox et al. 1988 study, and you need PET imaging to actually see this, is that glucose use actually massively increases as well during this stimulation. So glucose actually tends to remain coupled to blood flow. There's some exceptions that we can talk about, but for the most part, the finding is that there's this hidden increase in glucose use, which isn't visible to the bold signal, but is fundamentally there as a part of the reaction to stimulation. And so if glucose is being used in excess of oxygen, the other place where you might see something like that is actually in your muscles during a sprint. You're familiar with this as producing lactic acid, but when you're doing it in a sprint within muscles, it's because oxygen isn't available. And so this is a backup energy fuel. In the brain, what's strange and what's energetically inefficient is that oxygen is actually plentiful in the brain. There's tons of it, but for some reason it's not used, and glucose use is favored over it. So again, bold activity itself is accompanied often by an unmeasured changes in glucose use, which you'd only see if you were using PET imaging. In the rest of the body, that sort of sprint-like metabolism is the last resort, but in the brain, it seems to be a standard response to sensory stimulation and cognitive activity. And so the first question that I want to start with, and the fundamental one that we're going to build off of, is why is it that brain regions sprint, we'll call it, or so-called sprinting, when active? And again, to cover this, we need to do a little bit of background as well, just on basic brain metabolism in the first place. So how exactly does glucose and oxygen metabolism work? If we were to have a cell, it doesn't need to be a neuron, but let's say this is a neuron. We have cellular cytosol, we'll have mitochondria, and we'll have a sodium-potassium pump. If glucose enters, it enters the first stage of cellular respiration, which is called glycolysis. This is going to take NAD+ into it as a precursor. It's going to produce 2 ATP, so cellular energy, and that's mostly going to be used to power the sodium-potassium pump. Products of glycolysis are passed along and are passed into the mitochondria. MAS is the malate aspartate shuttle, but it's not going to be super relevant for what we're going to talk about. Within the mitochondria, the second stage, and this is the important part of cellular respiration, occurs, which is oxidative phosphorylation or oxphos. And this is where oxygen comes in, is combined with these intermediary products of NADH and pyruvate. This produces the bulk of ATP that we get in cellular respiration and produces CO2 and H2O as waste. Excuse me. So again, oxphos is the powerhouse. This is where we get most of the ATP. Production from glucose is energetically efficient, and we maximize the ATP we can get per glucose. At the same time, we can have another pathway where those intermediate products don't go into oxphos, and they're instead turned into lactate, which is exported out of the cell into the bloodstream. And in this case, ATP production doesn't carry on to oxphos. We get 2 ATP for glucose, and this is energetically inefficient. And the reason why you would ever do this is because if you weren't, or the reason why you would do this in the body typically is because if there wasn't any oxygen that could be supplied, then you need some way to restore the precursors to continue glycolysis. And what glycolysis into lactate does is allows you to replenish those in the absence of oxygen. But again, oxygen is plentiful in the brain, and it's even oversupplied in the bold response. So what could be going on here? And so again, just to define terms, aerobic glycolysis, because this term's actually a bit confusing, means glycolysis exceeds oxphos in the presence of abundant oxygen. So it's referring to glycolysis in an aerobic environment. And when we talk about activation-based aerobic glycolysis, we're talking about aerobic glycolysis that follows experimental stimulation, such as in an experimental design or in bold fMRI. And another thing that's important to emphasize is that this is inefficient, but it's locally inefficient, because that lactate that's sent into the bloodstream can be reconverted into pyruvate and NADH downstream, and then can be used in oxphos elsewhere. But part of the mystery is that all of this carbon that could be used to generate ATP is being exported from local areas. And another question might be, where else does this aerobic glycolysis occur? We're talking about it in activation-based aerobic glycolysis. It's actually fundamental to cancer cells, where it's been called the Warburg effect, and it's still a fundamental mystery. But basically, cancer cells just seem to gobble up glucose and then not use any oxygen with it. It occurs in red blood cells, which actually lack mitochondria. And it also occurs in the immune response, where it is thought to support biosynthesis. So it tends to be involved in creating cells to combat infection. But it's been ignored largely in brain imaging, or at least, if not ignored, it's not a very common topic that people talk about. And so why would that be? One reason that I want to suggest is that there's been a disconnect between neurochemists and neuroimagers. And there's been hypotheses that have been dismissed by neurochemists, but that have continued to be accepted within neuroimaging. And essentially what's happened is people thought that this was a solved problem and that they couldn't worry about it, but you maybe do need to worry about it. It's maybe something that is still an unsolved mystery, sort of in the center of neuroimaging. And so again, some common explanations that have been given for why aerobic glycolysis might happen during the bold signal is that maybe it's a fast source of energy. So for Marcus Rakel here, aerobic glycolysis produces ATP much faster, meaning that when speed is important, one might posit that aerobic glycolysis is the way to go. Aerobic glycolysis supports biosynthesis is another idea. So again, from the same paper, that aerobic glycolysis can remain elevated for an extended period after task performance, consistent with the hypothesis that it is in part a signal of experience-dependent biosynthetic processes in the brain. Or another idea is that aerobic glycolysis is localized into astrocytes alone, and that it supports glutamate recycling. And this is maybe the most dominant idea that a lot of people, if you ask them in neuroimaging who know about this, would say they think it has something to do with astrocytes. And again, from Marcus Rakel and Minton, the astrocyte, the synthesis of new ATP appears to be done by glycolysis alone. But there's evidence against these, so generally, as far as aerobic glycolysis being a fast source of energy, in terms of gross energy, that's not actually true. During activation, a slight increase in oxygen use actually generates almost all of the energy. So it's a very bad source to get energy quickly. There's some nuances to that, which we can get into later, but it's generally not considered to be the case by neurochemists to be a fast source of energy. The idea that aerobic glycolysis supports biosynthesis, well, it can be technically correct. Part of the mystery is that lactate is removed from the area. And if it was biosynthesis, then that would be trapping carbon in the local area. So that actually can't be the answer either. And then as far as the idea that aerobic glycolysis is localized to astrocytes, evidence has shown that astrocytes use OXPHOS against some of the initial assumptions of this model. Aerobic glycolysis also occurs within neurons. More recent work has demonstrated this pretty conclusively. And then even more review work recently has shown that if you total up all of the sources of OXPHOS and glutamate cycling, there's a one-to-one relationship between them, which shows that it's more likely that it's oxidative phosphorylation supporting this glutamate cycling. So you may find some of these irrelevant. I just want to knock these off the table so that you can trust me when I say that it is actually still an open question, because I've gotten pushback on reviewers because a lot of neuroimagers think this is all solved. Why are you talking about this? So again, to come back to the core mystery here that I just wanted to set up, most ATP, so most cellular energy, is being supplied by OXPHOS, but during activation, brain regions metabolize glucose inefficiently, as in they use glycolysis and then waste lactate out of the local area. Why is that? Why would that actually be useful from a brain design perspective? Again, keeping in mind this Darwin's dictum here that generally individuals are not going to waste nutriment on a useless structure. There should be some reason for this, and it's still an open question as to why. So the answer I want to give here, which we can then build off of to some other interesting questions, is that aerobic glycolysis is actually really well suited to provide cellular energy when and where OXPHOS wouldn't be able to satisfy the requirements. And the specific answer to that is going to be in ultra-thin axons that wouldn't physically fit mitochondria, but that maybe have purposes that are going to be relevant for us. And a secondary answer, or part of this answer, is going to be that aerobic glycolysis can promote informational efficiency, as in it can maximize the information sent per unit of cellular energy in these ultra-thin axons, but that it does that at the expense of energetic efficiency in terms of maximizing the local ATP you can extract from glucose. So again, the answer to this question that we're setting up is going to be that it supplies energy when and where it's needed to maximize informational efficiency. Okay. So one thing to highlight here as we start to delve into this answer is that brain design itself implies some certain trade-offs between properties. So one trade-off that's of interest is that transmission speed, in terms of how fast a signal can be sent, is going to trade off against energy consumption. Another is that information rate, in terms of information sent per second, is going to trade off against informational efficiency. So within the first, it's of interest that sending signals quickly over a distance is costly. You can actually think of action potentials themselves, which often people think of as a sort of unit of information in the brain, are themselves only one way of sending information. They're actually a compromise against chemical diffusion. You can send information faster, but you send it at a higher cost. So an interesting example of this that I really just couldn't help but highlight is that within C. elegans, there's actually not the genes expressed to even don't express genes for sodium potassium channels. So essentially, C. elegans have sort of foreclosed on the need for action potential signaling and instead perform it simply by chemical diffusion because they're so small that information can travel in that way. At the same time for us here, this trade-off between information rate and information efficiency is especially relevant. Axon diameter is going to be relevant to this. Essentially, thicker axons are going to have a higher information rate, and thinner axons are going to be more informationally efficient. And so this is a simulation study here using the giant squid model. But what you can essentially see as you go from left to right is that as axons get thicker and thicker, the information efficiency in terms of bits per ATP is going to go down. And I'll go through the details of why this matters here. So part of the reason for this that's going to be really important is that repolarizing after an action potential is going to be energetically cheap when you're dealing with a thin axon. So again, if you just think in terms of the biophysics here, if there's an action potential in a thin axon and a thick axon, and if the sodium has to be pumped back out again, the smaller the volume of the axon, the less sodium has to be pumped out before you can repolarize, because there's just less space and less need to sort of, essentially the resting potential is going to be more easily affected per sodium pumped out. So again, smaller volume means pumping out sodium costs fewer ATP to repolarize. However, the flip side of that is that channel noise is actually going to increase as the diameter shrinks. So if we had a thick axon and we had some random flickering noise of sodium getting out of the axon, then nothing happens. In a thin axon, it might trigger an action potential from just any errant potassium floating out of the axon here. So again, random leakage of potassium is more likely to trigger action potentials. And the implication of all of this is to say that axons can be made thin and they can be made informationally efficient, but that can only be done to a point. There's a certain point where you're going to get axons that are so thin that the noise in the channel is going to completely overwhelm any ability to send any information along them. And again, by simulation and then by sort of empirical data backing that up, that noise limit tends to be 0.1 micrometer. And so here's the empirical data backing that up. Parrish et al. Looked at a number of different axons in guinea pigs and just took stock of what the distribution of axon diameters were that were observed. And so what you can see here is that the diameter tends to vary. There's a nice distribution, but it tends to scooch down toward the noise limit down here. So you can see they tend to be quite thin. You'll get some that are much thicker, but there's a distribution bias toward these very, very thin axons. And what's important and what's really, really critical here is that we can also look, and these same people looked at this, along those same empirical axon diameter dimensions, we can look at the mitochondrial concentration, which, and again, mitochondria are going to be necessary for oxphos, for the cellular powerhouse. And what's extremely interesting here is that the mitochondrial concentrations as a percentage within these axons tend to drop pretty precipitously once you get into extremely small diameters. So if we were to line up to the same rate here, we can see that most of these distributions here are well under this area where you start to get a mitochondrial drop-off. And so again, the question might be, why would this mitochondrial concentration drop? And what's been suggested is that mitochondrial are taking up space. If they were in thin axons, you might be fitting them in, to steal a quote from Parrish here, like a pig in a boa constrictor. You're essentially having a factor to fit the cellular energy powerhouse into this axon and potentially thickening it, which again, if you have all of these advantages of having a thin axon, is going to work against you, and it's going to make it informationally less efficient by fitting the mitochondria into it. And so the sort of conclusion that I want to draw from this first part here is that oxphos itself, or oxphos, the cellular powerhouse, requires mitochondria. Glycolysis doesn't. And fitting unnecessary mitochondria would potentially thicken these axons and reduce their informational efficiency. So what we wanted to propose is what we're calling the efficiency trade-off hypothesis, saying that we can trade off local energetic efficiency for informational efficiency. And again, locally inefficient because lactate can be used elsewhere, but essentially the point is that glycolysis is filling a niche that oxphos couldn't. It could potentially fit into these extremely small axons and basically power or provide energy where it's needed in order to get the benefits of informational efficiency from these small axons that we know exist based on these distributions here. So that's the where, but I also wanted to touch quickly on the when as well. And so another property that people have pointed out when they've been thinking about glycolysis, and this relates to the idea of thinking it's a fast energy source, although it doesn't provide net energy very quickly. But there's a thought that glycolysis might be able to scale ATP production on and off relatively more quickly than oxphos. And there's actually an interesting analogy that can be drawn here that had been drawn by others with relation to the cancer research, that we can essentially think of oxidase and glycolysis kind of how we'd think of a city electrical grid. In a city electrical grid, what you tend to have is you'll have some baseload demand that's being satisfied by coal or nuclear plants. There's predictable variations within a day where we scale up or down energy generation, and we'll call that intermediate demand. But then there also tend to be spikes, like when there's a heat wave, everyone turns on their air conditioner at once. And what that tends to be satisfied by in efficient grids is by what are fuel efficient but responsive gas-powered turbines. And within neurons, we can think of something similar. We can think of oxidase as perhaps providing some baseload demand, scaling up or down to meet some intermediate demands. Mitochondria can be relocated as well. But if there's going to be rapid on, rapid off demands, glycolysis could be well positioned to fuel those by essentially helping match energy production to energy demand when it's fluctuating or when it's unpredictable. That unpredictability is going to come back to play a part when we talk about interpreting the bold signal in a moment. So again, functionally, these peaking generators are well suited to fuel unpredictable spikes in metabolic demand. Cool. So to sum up the first part here, the efficiency trade-off hypothesis that we're talking about is this idea that aerobic glycolysis might help to satisfy energetic needs without compromising the biophysics that help to promote information efficiency. Or it could quickly respond to rapid on, rapid off energetic demands, i.e. unpredictable demands. And a consequence is that the full ATP yield from each glucose is not going to be achieved when using this glycolysis route. But this is a local problem as waste lactate can be recycled and reused elsewhere. So we're not in a global sense losing energy or wasting energy. It's just local areas that are activated are spitting out this lactate to be used elsewhere. They're not making full use of it locally, but this helps solve other problems. Okay, so with that fundamental down, I'm going to move on to something that might be of more interest or more applicable. How do we interpret stimulus-evoked bold activation? And so if we're talking about activation-based aerobic glycolysis, what I want to sort of turn the question to is thinking about what activation means in this context in the first place, and if there's maybe some experimental designs that we've carried with us that have colored the way we interpret our data. So again, how should we interpret stimulus-evoked changes in brain glucose metabolism? And what I want to conclude and argue here is that we might want to actually think about abandoning the stimulus response paradigm that we may be very used to and consider activity in a prediction error evoked perturbation to the dynamic systems. We want to think about the brain as an ongoing dynamic system, and we want to think about what we're actually doing when we're delivering random stimuli, random experimental designs to people. And I'm hoping you guys will be sympathetic to this because you work in VR and you're used to creating actual naturalistic situations for people rather than bombarding them with randomized stimuli in the way that a lot of people are used to doing. So again, what does it mean to say that a brain region is activated? To come back to this figure here, what do we mean when we say that we've delivered a stimulus that we're going to see a bold response to afterwards? What's that stimulus actually doing? In real terms, we can say it's triggering a relatively small local metabolic shift, which again represents a slight increase in oxygen use and a large increase in glucose use through aerobic glycolysis. But is that a local metabolic shift representing processing, or is it representing something else? And I want to say we've been sort of let off easy by assuming that there's some sort of processing that the bold signal just simply stands in for. It's maybe something a little more complicated. So just to contrast a typical stimulus-evoked interpretation with the more predictive approach that I want to talk about here, if we think about typically when we show someone a stimulus, we'd imagine that it's taken in and then it's passed up a cortical hierarchy, and there's some processing that's done at each step. So the signal's taken in as an input, they're transformed by some sort of process, and they're passed up as a more complex representation. So we talk about ventral steering or ventral we talk about multiple visual processing streams and where there's some sort of elaboration and processing up a chain. And again, in this context, we think about activation as representing some sort of local processing that's going on. And so there's a predictive coding perspective, which I'm not sure if you guys are familiar with this or not, but I'm going to review it briefly. The idea here is that essentially what the brain has done is it's accumulated prior experience, and it issues predictions to predict that input. The idea is that those predictions filter the actual sensory signals that are coming in. So unpredicted input or prediction error is passed up the cortical hierarchy. And so what's happening at each stage is a filtering by top-down signals, and that filtering is happening through the entire brain and accumulated experience. So again, in this case, activation might not be quite local processing in the same way. We might think of it maybe as something like filtering or prediction error encoding. And again, there are many, many people who've been talking about these predictive coding models. Carl Friston may be one of the first most among those, but many other people have different models as well. And just to give a sort of core demonstration of this, which I really like, which my advisor, Lisa Feldman-Barrett, has used. If we look at this picture here, and if you haven't seen it before, it may look completely unfamiliar. And based on just the raw sensory signal that you're receiving here, it's hard to interpret what it is that you're actually looking at. But if we show you for a moment here that's a snake, and we look at it again now, it's actually very easy for a visual system to make sense of what it is that we're looking at. And in that case, we're using prior experience to essentially shape and categorize ambiguous sensory signals. So all I'm using this to try to demonstrate here is that it's not just a matter of taking in sensory input and transforming it. There's prior experience that has to be brought to bear, which sort of filters and categorizes that sensory information as it's arriving. So again, if we're thinking from a classic computational view, we might ask again, what is it? Why is it that this aerobic glycolysis, or this idea that activation is glucose inefficient, why is that so confusing in the first place? And you might say, if you're working off this, there's an implicit assumption that when a brain region is active, it's performing some sort of process. And so why would processing, if what the brain does is process, be glucose inefficient? It would, if you're interpreting the bold activation or these signals in response to sensory stimulation as fundamentally signals of the brain doing something, as opposed to not doing something at rest, it seems confusing that the brain doing something would be inefficient. And again, what would make aerobic glycolysis less confusing from a predictive coding perspective? We can think about processing isn't happening all at once. These top-down signals are a part of the local processing, and processing during the activation that we're seeing may be part of a larger process. It may be incorporating and filtering sensory signals into a larger part of the processing that has happened previously, predictions that are being passed down the line to local areas. And so what I just want to set up here as well is that we can use some of the links that we'd already established. So this link between aerobic glycolysis and small diameter axons, which we're calling the efficiency trade-off hypothesis, and the links between aerobic glycolysis and BOLD. And we can think about how to link across levels of analysis, specifically to prediction error and predictive coding over here. And one of those ways is there's actually links through norepinephrine. There's links through gamma oscillations, which are related to BOLD, norepinephrine, aerobic glycolysis, and small diameter axons. And then we have links through several of these over to prediction error as well. I'm not going to cover all of these here. You're going to have to look at the paper if you want to learn about norepinephrine, but we try to exhaustively go through all of these and establish the links to show that there are relationships between these here. We're just trying to holistically pull together a lot of neuroimaging measures into sort of one framework. But first, what I just want to do is walk through and explain gamma band oscillations and their relationship to small diameter axons, aerobic glycolysis, and BOLD. Am I doing okay for time, by the way?
Yeah, you're doing great. You've got about 20 minutes, I guess, left, but
okay.
How much you wanted to talk about, so yeah.
Perfect. Okay. So first, just to talk about gamma oscillations, I'm assuming most people are familiar, but if you're not, when we take EEG oscillations, we can divide these up into roughly certain bands. Gamma oscillations tend to be bands at higher frequencies, so 30 plus hertz. And again, there's a correlation between gamma and bold. Gamma band tend to be produced by fast spiking interneurons or their interactions with pyramidal cells. And another interesting fact of this is that gamma oscillations are actually glucose dependent. Recent evidence has shown this as well. So just to set up, briefly walking through these, and again, we can look at the paper for more details. I don't want to go through this exhaustively. I just want to sort of demonstrate that there are some links here. Work has shown that the bold signal tends to correlate quite clearly with positive bold signal. Specifically here in the 60 to 80 hertz, you can see along the y-axis a positive and negative correlation between bold and EEG. And interestingly, there's negative correlation between bold signal and slower frequencies in electrical oscillations, specifically alpha and beta bands you tend to see. And slightly theta bands, the extremely, extremely slow ones, you start to see some positive correlations with bold there as well. EEG oscillations themselves are produced. Bizaki and Wang have an excellent review pointing out that gamma oscillations are produced by interactions between pyramidal cells and fast spiking parvalbumin positive interneurons, which themselves are extremely branched, very thin axons. We're showing the axon shaft diameter here for parvalbumin positive interneurons, which again tends to be down in the like 0.2, 0.3 range of micrometers. This is again getting near that lower threshold, that noise threshold. And certainly well below in other cases where we were seeing a drop off in mitochondria. Another feature of interneurons that's important is that they fire at extremely high frequencies and they don't habituate, which others have pointed out is going to induce an extremely high ATP cost. So we can think about the fact that we have both an extremely energetically costly signal that's not habituating, firing at high frequencies, and it seems aerobic glycolysis would be something well suited to fuel something like that, if the efficiency trade-off hypothesis we were proposing is right. Okay, and then finally, EEG oscillations, recent evidence has shown, are actually glucose dependent. So again, lactate skips that early step. It can go right into oxphose. But if you give lactate, or if you give a bath that has no glucose in it, you'll tend to get suppressed gamma oscillation. So if you look, specifically, if you're looking at the bottom left panel there for the lactate, what's happening is slices are being bathed in a lactate solution, and it doesn't seem to be the right kind of fuel to fuel these fast oscillation signals at the gamma band. So again, there seems to be there's something in gamma oscillations that glucose is specially fueled to process. Or is a particularly fueled process. Okay, and then finally, just to link between gamma band and prediction error and small diameter axons as well. There's some evidence for bottom-up signaling, prediction error, and small diameter axon properties. So again, there's extensive work. There's been a lot of work on gamma oscillations encoding bottom-up signals, while alpha and beta encode top-down. What you're looking at here is a Granular causality graph, where essentially in a simple visual task with primates or in monkeys, V1 activity was able to predict V4. You can see at up in near the 100 hertz frequency range there. Essentially, gamma oscillations at the earlier part of the visual hierarchy are predicting gamma oscillations at the upper part of the visual hierarchy in V4, but then vice versa, V4 alpha and beta band oscillations are predicting their counterparts in V1 stronger than the other way around. So there seems to be some directionality in alpha and gamma oscillations. Again, in a bottom-up, top-down perspective. In a prediction error perspective, I can't go into the detail with all the experiment here exactly, but essentially, to summarize it really quickly, there's a really interesting study that's, or among a bunch of them, that are using simple and complex frequencies of musical notes, where essentially if the sort of simple sequence of notes is interrupted, you'll tend to get what we'd call a lower level prediction error, which is just an error in an error in sort of tone. If you have an error in sequence in a broader predictive sequence of sensory stimuli, then you'll tend to get a different pattern, also with gamma band oscillations, but in a different part of temporal cortex. And then the way that the variance was broken down this as well, there was also alpha and beta bands, which tended to predict these temporal predict as well in the other direction. And again, I should say that all of this is with intracortical EEG as well too. So it's at least not potentially scalp interference in this as well. Cool. And then for small diameter axon properties, a natural property of the small diameter axons is that because of the noise that we talked about earlier, that as they get thinner and thinner, they're going to have to fire at higher and higher frequency. There's more and more noise to overcome. Small diameter axons actually burst fire to overcome noise. And so they're actually well suited to infrequent high frequency firing. And this is the profile that we attempt to see in them as well too. Which again, if we're thinking about handling something in terms of prediction error, we're looking at something that's infrequent, requires a burst of activation, and then is quiet for most of the time. Okay, so if you find that convincing, then what does this all mean for interpreting activation itself? So one question that you might actually ask is, if we're thinking about the brain as an evolved organ that should be efficient in the way that it's working, or should be efficient, are the randomized stimuli that we're delivering in most experimental designs an accurate reflection of the kind of information that the brain evolved to encode? And so what I mean by that is that until the advent of computers, it would be quite rare for you to have a stimuli pop into existence, disappear in a darkened scanner, and then show up in a completely random and unpredictable order. We're used to navigating real environments, having continuities between them. And so it's been argued that what we might be doing in experimental situations is essentially forcing the brain into an unnatural circumstance of prediction error heavy processing, which might be why we'd see activation in the sense that we do with BOLD or with this associated aerobic glycolysis in a lot of the experimental designs that we've set up. And again, this is just coming from the perspective that we should deal with the brain in a way that understands that it was built, it was designed to optimize its experience, its organization. So again, if we're looking at a graph of ATP that's being consumed, and if we're thinking about the fact that the actual metabolic cost of this aerobic glycolysis is very small, but the glucose consumption is large, and that the large baseload, again, of this 20% of the whole body's metabolic budget that the brain is taking up, makes up the bulk of this, the actual cost that the brain is taking on, then we might think about this activation-based aerobic glycolysis that's being indexed in some sense by positive BOLD as representing metabolic ripples on a baseload oxidative lake. And again, I'm trying to be a bit provocative here, but I'm just trying to make the point that the brain itself, if 13% of the whole body's metabolic cost is devoted to signaling costs within the brain, the actual differences that we see during BOLD activation are very slight, in part largely associated with this pretty strange metabolic process of aerobic glycolysis. And so when we look at it, we might be looking at this metabolic ripple on the surface of a deeper dynamic system. And when we perform some of the experiments that we're doing, we're essentially just skimming the surface of this overall metabolic cost, or overall metabolic cost within the brain itself. And that's not to say it's not important, because what's actually interesting about this is that we're skimming a surface, but we're skimming a surface that's an extremely glucose-intense consumption process. That's essentially the brain sprinting to perform this metabolic activity. But in terms of the absolute energetic cost within the brain itself, it's a very small percentage. And so again, from a predictive perspective, what we'd say is that most of the metabolic activity within the brain is probably devoted to prediction, but a lot of that prediction-based metabolic activity in the brain is probably invisible to some of the bold imaging techniques that we'd actually use. It's probably happening constantly, and it's difficult to actually image based on fluctuations, the perturbations of this dynamic system. So if that's all correct, then what does that actually give us? What it gives us that we can start to think about information processing, not just, start to think about information processing in metabolic terms, not metabolic terms, I'm sorry, that's a mistake. The idea is that prediction error may have metabolic glucose requirements that prediction doesn't. And if that's correct, then it's actually extremely interesting that we can start to split up channels of information processing and different metabolic consequences of each of them. And of course, I should say this all with the caveat that there's experimental work that's required to follow this up. These are all hypotheses from trying to link together a lot of work. And granted, I'm trying to work on right now is actually specifically trying to look at that and image glucose, oxygen, and cerebral blood flow, all at the same time using combined PET and neuroimaging. So again, I'm proposing this. It needs to be tested, but if it's right, then we might have some reason to think very differently about a lot of neuroimaging results that we already have in terms of what they're really telling us. Okay, that concludes the core metabolic part. And the last thing that I want to get to actually is the thinking about social motivation in metabolic terms here as well. So again, if we can think about information processing in metabolic terms, then we could think about prediction error in terms of glucose consumption, having an excess of demand for glucose consumption. How do we think about the expectations of other people and their ability to motivate our behavior in individuals? And again, the idea is that conforming to the expectations of other people can actually be a control process to help regulate prediction error in a social environment. And then the metabolic costs of that local social environment can be controlled through your own behavior. And so to sort of set this up, what I'm trying to grapple with is that human social behavior itself is actually something that varies drastically across individuals, cultures, and periods of history. There's a tendency in social psychology to look for universals in human behavior, but the reality is that it's extremely, extremely culture-bound variable, and it's probably going to change over time as well, too. So what I want to show in this last section here is that models of human motivation can be refined by grounding them in brain metabolism. And that this matters because having a different model can help us get a different understanding of how social behavior works and what that social behavior is actually aimed at achieving. And to do this, we're going to have to think a little bit about subjective experience and sort of phenomenology of motivation and obligation. So first of all, why does the subjective experience of motivation actually matter? And I'm going to give you an example where it doesn't matter, where people don't typically think about how it matters in the same way. In a typical prisoner's dilemma game, which people might be familiar with, people are faced with a dilemma where they decide if they're going to cooperate or betray a partner. So two people play this. Each person decides if they're going to cooperate or betray. There's a reward structure that's paid off with this. And there's solutions that come out of this, but there's solutions that are based on the reward structure and how people navigate it. So the traditional solutions here, and again, people draw lessons for actual human behavior from this, is that the short-term strategy is that you betray a partner and you maximize a reward. But if you're in a sustained social interaction, then you cooperate. And so the lesson here is that if you build up a reputation for yourself, or if you build up a reputation as someone who's going to cooperate, you maximize long-term utility. And that's building off of the assumption that it's going to be a sustained social interaction. But again, what I want to press is whether you really feel this way or whether you're really thinking very consciously about your reputation, if that's a subjective experience that you've had when you've actually cooperated with other people. What's actually motivating this normative behavior in real situations does the game actually feel the same as a real social obligation? And I'd argue that in a prisoner's dilemma, it really often doesn't. In contrast, social psychologists have studied social pressure and have studied conformity and social influence. And so this poor little nerd here in the middle is a participant in the classic Asch experiment, where a number of Confederates, in this case, 6 other people, would sit around a table, and they would all judge whether the line on the left matches the line on the right, which line on the right matches the line on the left. They'd go through a couple and answer correctly, and then you'd run in a situation where you would run down the line, and every single person would answer B. B, B, B for all of these guys all down the row. And then we get to our poor actual real participant here, and he feels extremely uncomfortable. And then many people would end up going along and just going with what the group had said in this case. So this is an example of people feeling an obligation or feeling social pressure to conform in a social situation. And again, there are other situations where people conform for other reasons. I don't want to say that people conform only under obligation. People can conform under informational influence, where you just copy other people because it's beneficial. But what we're really interested in here is this social pressure, this feeling of obligation where you behave as other people expect. And we call that normative influence. And again, often inspired from some of these behavioral economic models, people tend to provide explanations of why people feel motivated by expectation. They'd say, maybe it's reputation. So they might say social norms enjoin the behavior of others through a promise of social sanction, where you're going to punish people that don't conform. Or they might be transmitted through the punishment of deviance and the social success of followers. Again, I'd give a caveat here, which is that this is all saying that violating social norms or violating being a nonconformist is something that's going to get you punished, when actually being a nonconformist is pretty cool under a lot of circumstances too. So you can't really hold that up as the perfect explanation of all of this. And there might be some room to understand some nuance to how people really feel the social pressure. It might not be purely through reputation. And actually, Adam Smith gives another example of this that helps to sort of clarify that there can be multiple motivations at work here. Reputation doesn't really feel like normative social pressure. And he's argued that nature's endowed us not only with a desire of being approved of, but with the desire of being what ought to be approved of. The first desire could only have made us wish to appear to be fit for society. The second was necessary in order to render us anxious to be really fit. That anxious is actually going to be quite important because we want to tie this into the affective experience as well too, which is going to be a feeling of anxiety. What I want to say is that this sense of should and the metabolic processes that we've already reviewed are going to help us understand it in computational and biological terms. So again, what we've already covered in the earlier part of the talk is that from a biological perspective, if we think about the costs and benefits of behavior, if we really do have a link between metabolism through glucose metabolism and certain kinds of information processing, specifically in terms of prediction error, then what we do is we have a link between a real metabolic or real cost that we're aware of biologically and a form of information processing, specifically processing prediction error, processing uncertainty in a social environment, when things just aren't happening as you would expect them to. And what I want to really briefly go through here is just a computational sketch of how you might get something like motivation out of this. So we might think about what patterns of behavior do these brain level details actually facilitate. And then finally, we want to think about explaining variability. How do these patterns of behavior help us understand other social phenomena? So again, we've already gone through this idea of predictive coding, but if encoding prediction error actually has a glucose-based metabolic cost, then there's a cost that's under control of how much prediction error there is in the social environment, or in the environment or in the social environment specifically. And what we want to say is that this cost can be, or those costs, the metabolic costs can be controlled by controlling prediction error, which is information. So if those costs are reduced when an environment is predictable, but at the same time, you need accurate predictions. In order to make accurate predictions, you need to take up information. You require a powerful internal model. What we have here is an explore-exploit dilemma. So we can exploit an environment and minimize prediction error, or we can pay a cost, a glucose cost, I'm arguing here, to explore and then to build a better model in the future. And what I'm going to be talking about, or what a sense of should, or what this feeling of obligation is going to be, is specifically feelings of affect based around exploiting a predictable, creating and exploiting a predictable social environment. Okay. Specifically, what I'm going to say here is that conforming to other people's expectations can help you to regulate the metabolic costs and the affective experience of the social environment. So when you're predictable to other people, you can make other people predictable for you. So just to lay this out, if you have a brain and if there's a metabolic cost to you that's proportional to the prediction error you receive, that prediction error is just going to be equal to the predictions that your brain is making and the difference between the behavior of individuals in your social environment. And it's the same thing is true for other individuals. So they have some prediction error they're receiving or information they're encoding. They have their own behavior, or they have your behavior, and then they have their own predictions about your behavior. If your behavior were to massively violate their prediction, if I were to commit some really egregious social norm right now and just violate what anyone expects typically in a presentation, then what I'm doing essentially is I'm feeding you information. And that information is going to change your internal model. Information is prediction error. And if it's going to change your internal model, it's going to change your behavior potentially in some way. And the more that it changes your behavior in ways that run ahead of the predictions that I could actually make about them, the more prediction error I'm going to receive. And I want to say the more metabolic cost or glucose cost you'd actually receive. So if we reduce this all down, what we're just saying is that you experience a metabolic cost of prediction error, and that cost is proportional to the difference between your behavior and other people's expectations about your behavior, their predictions. And we could sort of pseudo-formalize this. We could try to clean it up. But essentially what we're saying is that you can exploit the predictability of a social environment using your current model by picking a behavior that conforms to others' expectations. An important aspect, and as you guys have talked about being interested in arousal or in affect, affective arousal tends to be associated with interoceptive information and specifically with prediction error. And so those affective and interoceptive consequences, interoceptive from the metabolic costs, can be learned, anticipated, and avoided. And so essentially what I'm saying is that when you feel a feeling of social obligation, what you've learned is that your environment will tend to be more unpredictable, more uncertain for you, and you're going to increase your metabolic costs when you violate people's expectations. There's a question that I'm not answering here, or there's an open-ended element of this that I'm not answering, which is how you actually figure out what people expect of you in the first place. And I don't totally have time to get into that, but we can talk about that later too. So essentially what I'm arguing here is that social pressure, which you'd experience as a sense of should or a feeling of obligation, is an anticipatory anxiety felt toward violating others' expectations. And then the conscious experience of that anticipatory anxiety is a predictive signal of real metabolic costs that you would incur by disrupting your social environment. All right, I'm going to wrap it up here very quickly. So again, how do those patterns of behavior, or this idea of your own conformity to social expectations being a way of controlling prediction error in a social environment, how can that help us understand other social phenomena? And what they do is they point to how metabolic regulation and interacting individuals can create and maintain complex social structures. So to come back to the simple example of a prisoner's dilemma here, to go through the traditional solution again, we're sort of given this dichotomy of how people solve this. We say that you either have a short-term strategy, which is to betray your partner and to maximize a reward. Or we have the long-term iterative strategy, which is to cooperate, build a reputation, and rely on reciprocity. So here, cooperation requires a cooperative and predictable partner. You need someone else who's going to cooperate back with you and isn't just going to keep betraying you. What this perspective would say about this really simple case of a prisoner's dilemma is that you actually have another possibility open, which is an ultra short-term strategy, which is to behave as other people expect. You literally are making yourself exploitable to other people in the hopes that this is going to stabilize their behavior in some way and cause them to behave predictably. Cooperation, if the cooperation was what people expected, can create a predictable partner. And it seems counterintuitive in that it's not going to work in a prisoner's dilemma game because the options are so few. It's probably not actually going to work in this case because you've basically, the whole reason why you would conform to people's expectations is to keep them from behaving really off the track or in unpredictable ways. But you just have a simple 2x2 of behaviors that people can possibly take here. So the fact that this is not a naturalistic situation kind of forecloses on some of the strategies that you could take. But if you're actually out in the real world, then there's a limitless variability and direction that people's behavior can go, and conforming to people's expectations can help put some constraints on the number of directions that people can behave. Okay, so again, thinking about this terms of feedback or social sanctions, or how behavior is shaped by feedback from others. If we have this diverse set of behaviors and cultures and customs that people can behave in, how is it that we get some sort of stability or some sort of set of social customs that come out of that? There's a really excellent point that Boyd and Richardson made. It's the title of their paper in Cultural Evolution that says that punishment allows for the evolution of cooperation or anything else in sizable groups. But what this perspective is arguing, which is that conforming to expectations can keep other people from behaving in unpredictable ways, and violating expectations can cause social uncertainty and metabolic costs, is that through an affective response to self-caused social disruption, your brain is actually punishing itself. When you violate people's expectations or when you violate norms, no one has to intentionally punish you. People are just reacting in their natural way to your behavior, and you've created a socially uncertain situation for yourself, and you've created metabolic costs from it. So there's a self-reinforcing nature to this here without anyone even needing to intend to punish you. And so culture itself in that way might actually be an emergent product of this runaway cycle of reinforcement. Simply by reacting naturally to unpredictable information that you're giving them, people are going to become unpredictable and exert a punishment on you to bring you back in line with social norms and expectations. And so intentional sanctioning, where people actually go out of their way to try to enforce the rules on you, might actually be the exception rather than the rule. And I know you guys are in New Zealand, but if you see the way that mask use works here, or with COVID-19, these different struggles about whether to wear a mask or not, I've had no case where anyone's really ever yelled at me or told me not to wear a mask, but there's certainly been cases where I feel uncomfortable. I don't know what the social expectation is in a given situation, and I just try to go along with the norm in order to minimize the likelihood that something unpredictable is going to happen. Okay, and then finally, this can help us think a bit about a different perspective of how social groups might be formed and maintained as communities of expectation. So there's been a proposal by some that humans have an instinct for tribalism. As a quote from Jonathan Haidt saying, "When I say that human nature is also groupish, I mean that our minds contain a variety of mental mechanisms that make us adept at promoting our group's interests in competition with other groups." There's alternatives though, and what this mechanism is doing is presenting one, which is that if people conform in order to minimize prediction error, then that works when they can model what's actually expected of them, which we didn't quite go over, but you need to know what the expectations are. You need a way of making an inference about what people's expectations are. And we could define a group as a set of people for whom you can estimate mutual expectations at some minimal cost, and they can estimate yours. A group is a community of people where we know how to behave around each other. If the costs of improving your estimates are high, if you're around someone where you really don't know what they expect, it would take a lot of iterative interaction with them to learn it, then you might self-select out of social interactions. And you could see social groups potentially form in some emergent way through this. And just another point as well with that, Dita Marie Munch-Jarrett, who's a philosopher who's worked on some of the ethical implications of work related to this is just making the point that anxiety, social anxiety around people that are different than us might play a larger role than antipathy. We're very, people are very interested in implicit bias or an explicit bias, but a lot of the time you might see social groups form or bias form just when people are uncomfortable with people that are different from them rather than having an active antipathy to them. Okay, so to close out here, just to come back to these quotes again, what I was hoping to get across in this talk is just that what we need is actually a paradigm that takes biology as fundamental to psychology, but also that shows a path forward to get, or also a paradigm that's going to have a path forward to get some empirical hooks into social psychology and neuroscience. So on the neuroimaging end, we talked about the bold signal and how it's a fantastic tool for measuring aspects of brain metabolism as people who are awake and who are able to report on their experience are able to think and behave in the scanner. But we can only really integrate that knowledge from neuroimaging into the larger project of neuroscience if we understand the metabolism that's really underlying it. And so what I've hoped is that this work can bring some clarity to that metabolism, and it can help to break us from some old habits of interpretation, like imposing a stimulus response framework onto a brain that might be adapted to act efficiently in continuous and naturalistic environments. And I also hope that on the social psychology side, even though I was sort of going through it very quickly there at the end, I hope that I was able to convince you that it's worth taking subjective experience seriously. And that when we make understanding phenomenological experience a genuine scientific goal, we can start to open up new avenues for dealing with social behavior. So again, just to close out, if we want to look at subjective experience, it's very easy to get lost in the subjectivity. But by thinking in biological terms here, we can help make this project really scientifically tractable. We can help to provide, as the last quote here is saying, a backdrop, a sort of biological backdrop on which we can think about this shadow show of human social behavior. Cool. And I went over time. I'm really sorry, but thank you guys for having me.
Great. Thanks so much for the talk, Jordan. There's a lot to unpack there.
Yeah.
I'm sure there's a few questions. There's a couple in the chat, actually. Maybe we can start with those ones. So Elisa was asking right at the beginning about the neuroimaging modality that was used to capture some of those early data. I'm not sure, Elisa, if you want to ask the question, you're online here.
Hey, Jordan. Thank you for a great presentation. It's awesome.
Thank you.
So I missed for early part of your presentation. So I was wondering, since you were measuring glucose, I was wondering, and also bold signal, I was wondering that you are using PET or fMRI or fMRIs.
Yeah. So I think for the slide that's relevant here, I'll share a screen so I can pull this back up again. For this here, for those little heat maps on the side. On the next slide is the glucose. This is PET. So it's Fox et al. 1988. It's a really classic study. It's probably like before bold was even really a thing. But basically it's done in multiple PET scans where the glucose consumption is with FDG, and then the oxygen is with O15 tracers. And I think the blood flow was with different tracers as well. So basically, these are all from different scans, but it's using the same very simple stimuli of just flashing the checkerboard up on the screen. And they're part of the reason I talked a little bit about why people ignore this like glucose consumption that happens during the bold signal. Part of it is that it's really expensive because you need to use like PET tracers to look at it. Or you need to use, I didn't talk about here, but specialized techniques like MRS or magnetic resonance spectroscopy. And that can get you a look at the relative lactate changes, which is another way to look at this glucose consumption. But these are PET scans. And one of the things that I'm trying to do in some work that I don't have slides to talk about because we haven't collected the data yet, but there's some ways to do combined PET MRI imaging. And so there's potentially some ways to get glucose and oxygen consumption all in one shot. which people generally haven't done, and they haven't done at the level where you're getting sort of absolute consumption for each of them. But yeah, long story short, the modality here is PET imaging for this.
Another question I have, it's about, so most of the things that you were using is, it seems that just except EEG part, which is from gray matter of the brain and also cortical region, most of it is comes from subcortical region. So, and I already guessed about, for example, probably you used like PET scan because you were measuring glucose. So most probably you were using PET scan. So I was wondering that since part of our lab, they are working on emotion recognition. So we are trying to hybrid system. We are trying right now fNIRS and EEG together to find out about brain emotional state and different situations, and make it possible to, instead of being in a constrained or expensive devices, we can use just something like portable devices like fNIRS and EEG to understand emotional or mental state of the participants. So I was wondering that if you have any suggestion about how we can measure mental state using fNIRS, because the reason that fNIRS, especially in our work, time is so important. So we, unfortunately, bold signal or in general, for example, glucose detection or tracing the glucose, they have a lag between what you want to do and then depends on which part of the brain or muscle, FNRE or FNRE, you are monitoring. It may take some seconds, like for example, 5 to 10 or maybe 15 seconds to be appeared there. Do you have any suggestion how we can cure this disadvantage of fNIRS or in general bold signal?
The lag time or how to get it to better line?
Because we have EEG, but EEG is not accurate and also it's not local. So it gives us it doesn't have spatial information at all. So, but it has temporal information. In other hand, fNIRS has spatial information, but it doesn't have temporal information. So we want to kind of combine together to maybe cover the weakness of other one. So do you have any idea that how or suggestion that how we can or is there any criteria, for example, to using one of them, like fNIRS, not just EEG, put aside the EEG, just using fNIRS, and in the same time, we get a reasonable temporal accuracy using fNIRS?
Well, so I think to go to your first question, though, just about how do you combine these, I think, so I mean, I know that's a project of a lot of people who try to think about how do you combine the advantages of these different methodologies, right? But I think, I don't know if I have something super unique to say to it, except just that the sort of core goal of what I'm trying to talk about here is that I think the real way for you to combine those methods or to actually make these things really work together is to have a more concrete understanding of the actual mechanisms that are underlying them. Like what is really driving the bold signal? What is really driving the sort of blood flow that you're measuring with fNIRS, right? And once you have a deeper biological understanding of what's really causing that mechanism, then it really can start to complement. It can start to complement because it's telling you some a real underlying empirical information that you can interpret in relation to other biological information, rather than it just being all sort of measures of activity in different ways that you compare and that you have different temporal and spatial resolutions for, right? So I think, it's a little ironic, I've talked about other people with this before, but it's a bit ironic that when we think about bold or fNIRS, because we don't have a total understanding of the biology that's underlying it, it's a quantitative measure that you can get numbers out of to make comparisons between conditions, but it's kind of a qualitative measure in that you don't have a real fundamental understanding of what it's really showing you. And I think that's honestly a bigger problem than even the temporal and spatial resolution issues. It's that you're essentially looking at a qualitative contrast between conditions and seeing that something is different. But the way that you interpret that is going to be really colored by a lot of the assumptions that you bring to bear on it. And what I was trying to talk about here is that those assumptions that people bring to bear are assumptions of a stimulus response model, that what the brain is fundamentally doing is reacting to and doing processing in response to the information that you're feeding it, right? As opposed to the brain being a real dynamic system that's coupled to its environment, and that when you give it conditions in an experiment, you're sort of perturbing that system and throwing it out of its normal dynamics, right? So I'm not totally answering your question, but I'm sort of dodging it to try to just underline that, like,
No,
I
thank
think
you.
it's a biological understanding that you got to have. Yeah.
Yeah, thank you. Thank you. It's a great presentation, Bo.
No, thank you.
So we had another question from Cornell, who actually had to jump to another meeting, but he wanted to ask me to ask it so he could get your recorded answer. And so he's asking, what is the stimuli used in your experiments for bold activation, and any insights on EEG or bold activation for emotional stimuli like video, audio, or images, or VR maybe?
So I think, I'm not sure which experiments, if it's the core sort of bold activation, the stuff that I was drawing from there was just the classic Fox and Rakel work, where it's just a checkerboard to get some visual stimulation. I'm not sure which other stimuli, but if you want the details of it, you could probably look at the paper.
Sure.
Just give me a day to put up a more recent draft of it, because I've got an edit to sort of improve it a little bit. But the as far as insights for EEG and bold activation for motion stimuli, I mean, I think I don't know if I have a good answer, but I do want to say that I think it helps to think. It can maybe help to think about emotion stimuli in a broader sense as well too, because part of what when we're talking about affect, or just people's affective experience, part of what we were talking about with the sort of the social component of the talk was the idea that that uncertainty itself, or uncertain social situations, is going to be an affective situation to itself. And so yeah, I'm not sure if I have insights except what we were already just talking about, though.
Okay, well,
so sorry, I'm trying my best.
I mean, I know that the second part of your talk was largely theoretical in some ways. I was interested as well in terms of, you're talking about a metabolic underlying source for the need to conform to others, and I'm just wondering how you would be able to run experiments like that. Would you run some of the similar experiments that done in the 50s with, peer pressure experiments, but this time be able to use EEG or other imaging to record brain activation during those experiments? Or how would you be able to conduct experiments that would be able to look at the metabolic processes?
Yeah, I mean, so I think I want to work my way up to looking at the metabolic processes, because I think the actual methods that you have to look at metabolism in a dynamic metabolic cost in a real social situation is maybe tricky. It's maybe a case where the theory is going to have to run ahead of the actual empirical data you can do for a bit, just for methods reasons. But I think the place to start with it is to look at the affective component of it first, and specifically to look at arousal. Mm-hmm. Because we know that arousal is going to be associated with prediction error, and the extra leap that we're making on top of that is that the prediction error is going to be associated with a metabolic cost. So some of the designs that I'd like to do are things like looking at the way that people regulate themselves in social interactions, even in maybe subtle ways. Like I'm super interested in eye contact, especially. So how people in social situations will sample other people's faces, and how that might co-vary with ambient arousal levels, and whether people might sort of sample the dynamics in a way to keep themselves at a reasonable arousal level. because I know that there's some work, and it's really interesting work on predictive coding and autism about essentially people with autism being potentially hypersensitive to prediction error, or having a disrupted balance of signal to noise in terms of encoding information, that there might be affective consequences to that. And what I'd like to see in normal situations is, yeah, how people, when they're engaged in a face-to-face interaction, direct their gaze in ways that's either going to be sampling parts of the environment that are going to be noisy, or largely predictable and stable. So for that, you probably want to hook people up to physiology equipment, and you want some eye tracking glasses, ideally, or something to engage in social interaction. But yeah, that would be on the social end of it. And I think to get at the metabolic piece of it, for a while at least, I'm probably going to need to rely more on sort of scoping out a reasonable theoretical paradigm and tackling some of the like bits and pieces one by one, because it might be tough to do it all in one go. But there's different parts of this that we could prove with different experiments, I think.
Interesting. Yeah, very interesting. Does anybody have any other questions for Jordan?
I have a question, Mark.
Yeah, go for it, Aaron.
Thanks, Jordan. Thanks for the talk. It's really fascinating, and now understand the biological side of the things that we never considered that much. So my question is on a very high level, right? I'm pretty curious to know, now that we understand this unpredictable situations in social settings has a metabolic cost in terms of coding. So, so do you think that this, we can kind of use this, say, let's say we can use VR and put people into an unpredictable situation where things happen out of the norm? And can that help people who are, let's say, diabetic, who have issues with the glucose metabolism? So can it treat people in some way?
For the glucose, or for the, interesting. I don't know about that. I mean, it would be, what would be interesting to me would be to see how people, I mean, it'd be interesting to see how people who are hypoglycemic, how they observe social norms, or what they do in social situations. Like, I mean, that would be really interesting. I don't know if you'd run into, if I'd say that, you know, sensitizing people to norm violations is going to help them deal with the biological consequences of hypoglycemia. I think it's more going to be the other way around, that like, if there's a severe metabolic disruption, it would be useful to see how people regulate their information uptake in like an explore-exploit sense, right? But you got to we'd have to get more specific to try to test some of this than it just being general explore-exploit. But the fundamental principle of it should just be that there's that there should be a glucose-related cost to prediction error. One thing that I should say, though, that's actually really important to clarify, is just that it's really easy with this work, and I didn't say this explicitly, to get it mixed up with there's the classic work in social psychology that's completely failed to replicate this Baumeister stuff about glucose consumption being associated with glucose being consumed with self-control. So the idea that, you know, if people like are low in blood sugar, then they're going to be like not engage in control in the same way. It's this ego depletion work that's failed to replicate. I think the problem with that, and the part that makes it actually difficult to test directly some of the stuff that we're talking about with glucose consumption and related to social norms, is that actual circulating blood glucose is going to be a pretty well-defended parameter. And so if you think about the body as, or the brain as an organ that exists to regulate the body and to sort of regulate the organs and systems within it, it's not good for you to have wild fluctuations in blood glucose according to just social interactions and things that are going on in your environment. And so honestly, you don't expect to see massive amounts of variance on those variables. What you'd expect to see is at a local level, some of that glucose consumption that we observe in the brain when we use the PET scan, right? But you'd also expect compensatory systems in the body to like regulate that, such as insulin as well, right? To bring blood glucose back up again and to help regulate that parameter. And so that's regulatory systems within the body. Behavior itself is also going to be one of those regulatory systems, right? So what I'm just trying to sort of add to that is to say that, you know, people's decisions to engage in explore versus exploit behavior are themselves going to have metabolic consequences, and they can be part of that system to try to regulate these variables at certain levels, right? And I think we didn't talk, or I didn't talk about it in the talk, but in the aerobic glycolysis paper, we have an example that I really like that relates to how some of this might be relevant for depression. Specifically because there's been a lot of work looking at the metabolic consequences of depression, or the sort of metabolic features of depression. And it's frequently associated with metabolic disruption, but it's also associated with essentially chronic inflammation. People are sort of chronically have a hyperactive immune system. Mm-hmm. And what's important about that, and why I'm going off about it, is because it was mentioned offhand earlier in the talk, but the immune system is actually one of the other major sources of aerobic glycolysis, apart from cancer or red blood cells. And if the immune system is scaling up activity, then it's actually going to require glucose through aerobic glycolysis as well, at about the same rates of what the brain consumes in terms of total metabolic resources within the body. It's around like 14% or so. And so if you had if you basically have a chronic increase in the immune system, then you're going to have a sort of steady increase in glucose demand for the immune system. And there's been other work that's been proposing that that essentially the cytokines that are released from that chronic inflammation travel up the vagus nerve, go to the brain, and interact with dopamine systems to regulate against exploration behavior. And so if that's all true, then what you might be seeing there is essentially another way to understand what's going on is that you have an increase in glucose demand in the periphery in the immune system. And then you have systems within the body that are interacting through cytokines, the dopamine system, to regulate behavior, bring down exploratory behavior, and dial back the amount of glucose that's going to be required for encountering novelty and encoding it, right? You essentially get sickness behavior and depression where you try to avoid doing things that are going to be extremely novel and potentially taxing on this prediction error encoding. So it's, I guess, yeah, long story short is like, it's really complicated. I don't think there's like easy slam dunk tests for a lot of the stuff that's in this model. But the sort of thing that I'm trying to propose and I'm trying to get across is that you need a paradigm and you need a way to sort of link up between a lot of existing observations and find a way to think about how they work coherently together. And metabolism is a really, really nice paradigm to do that with because it can connect to a lot of things. And because there's fundamentally like really important metabolic aspects to the way that neuroimaging works, which have not really been, they haven't really been fully understood to the extent that they could be. And once you do really understand those, it opens up a lot of potential for developing this in a really rich scientific way, rather than in the sort of qualitative way that we have brain imaging work right now.
Great, thank you so much. Yeah, it's super complicated. I do understand. And there have been some other research as well that connects gut health and metabolism to autism as well. So yeah, I guess we can definitely use this understanding, both our neurology and our metabolism, etc., to treat various different kinds of problems that people have or disorders that people have. So yeah, no, great. Thank you so much for your talk. Thank you.
Oh, thank you.
Great. Does anybody else have any other questions? I had one other question for you, Jordan, I was really curious about your comments about trying to reduce the activation energy basically between, your activities versus others, and how unexpected input may cause increase. So in our lab, we've been doing some work on, we talked before about hyperscanning and synchronization of brain activity. And I'm also interested in physiological synchronization as well. People will have similar heart rates sometimes, or their walking rates may get similar. And I'm wondering if that also plays into what you've been talking about in terms of reducing the predicted versus actual metabolic cost. And so do you have any thoughts about that and what we could be looking at in terms of trying to look at areas where you get that synchronization happening as well?
Yeah, no, totally. I mean, I think that
one example is what you're talking about just at the end, what you're talking about before about the gaze. That's very interesting for you. It seems like if you can get in a situation where you've got a lot of gazing going on and you're looking at other people's faces, then you get some expectation around their arousal level, and that may synchronize your arousal level with their arousal level, and then also minimize that cost as well.
Right. Yeah. So, I mean, there's the questions of like, what's the target arousal level that you're going for, and whether you're trying to synchronize, whether there's physiologically a sort of optimum that people seek when they're trying to synchronize arousal between each other, when they're trying to synchronize behavior. I'd be really interested in whether, what are the sorts of, in terms of absolute metabolic activity, like what is so there's a question of what's the bold signal going to look like as you get more and more synchronous in behavior in a domain. And my guess is going to be that asynchronous behavior is generally going to increase the bold signal.
Mm-hmm.
That's the interpretation, if that's representing some sort of prediction error, is that as people get more and more in sync, you should get fewer perturbations from a sort of ongoing dynamic state. So you should have less and less need for sort of prediction error and associated bold signal. The bold signal, the other thing I should say is the bold signal itself it's still going to be a little complicated, because I wouldn't go so far as to say that the bold signal just is an indication of is prediction error. It's more of an index of this aerobic glycolysis, which I'd be more confident saying is something related to prediction error, but there's really important cases where they pull apart. But I'd still guess that, as you get more and more synchrony, you're probably going to get fewer perturbations and fewer sort of fewer canonical bold signals. But yeah, what sort of synchronization do people able to do in the scan? I'm really curious about that, actually. From
what activities, or what sort of synchronization?
What sort of activities?
There's been a wide range of activities studied. In our lab, we've been doing work on finger tracking. We have two people sit opposite each other, and they start moving their fingers around, and one person follows the other person's fingers. But you measure brain activity before and after the activity, not while the finger tracking's happened. And you find that for certain bands, you get phase synchronization between after they've completed the activity. But there's been other examples of people using synchronization in card playing games. So you've got four people playing cards, and there's two pairs of partners. Sometimes your brain activity will synchronize with your partner and not synchronize with the opponent pairs.
Interesting.
And the people have also looked at economic games like trading games, or the Prisoner's Dilemma games. So a wide range of activities, actually. So go on.
Oh no, it's just reminding me, there's a study that I really like, but I'm not sure if it's published, but it's by a friend of mine, Bo Sievers, that was looking at synchronization in, I think he had groups of people watch a sort of weird abstract movie that didn't have a plot or make sense, but it was a sort of sequence of scenes and bizarre art house sort of thing. Had people watch it individually and then get outside of the scanner, discuss it with each other, and come to a consensus of what they thought it meant, and then watch it again. And then they had more synchronous activity after that, and independently synchronous activity with each other compared to other groups of people that had all watched it and come to a different consensus.
That sounds really interesting. And I'm not surprised. It's been, yeah. So I'm just curious with what you've been talking about with the prediction. I guess as a group, people might try and minimize the variance or the activity that's unexpected, and so you may get a tendency towards more synchronization too. Is that what you're trying to say?
I think so. So I mean, I think that if they were watching them, so just to start it with the simple example, if people were watching a movie over and over again, like essentially part of what I'm saying with the bold signal probably reducing and being related to prediction error, it's another way of just talking about a classic habituation effect as well, right? Like and people have used sort of predictive coding models to try to explain just what you get of habituation, of decreasing intensity of signal when you repeat something over and over again. And so if you are watching the video repeated times, you'd expect to get less intense activation on repetition. And so, yeah, I think in a situation where you are having individuals synchronize behavior, if they're sustaining that synchronization, and if they're receiving feedback, I guess also, what sort of feedback they're receiving about whether they're in sync or not is also probably going to matter, right? Like, I think it would depend on the task a little bit also. I'm trying to think about how it would work with the synchronizing fingers across there. Did they get feedback? Could they see that their fingers are synchronized with the other person, or?
They can see that, yeah. Well, there's different variations. But yes, they can certainly see the fingers. Sometimes they can't see each other's faces, but yeah, definitely. But yeah, they can definitely see the finger movement.
Cool.
Yeah. So does anybody else have any other questions? We're kind of out of time, and we've had some. Thanks so much for an interesting presentation. But it's a great opportunity for others to ask questions. I'm not sure. Anybody else have any questions? No? Well, let me thank you again, Jordan. It's been a really fascinating talk. And as I said, it's been a bit different from some of the previous topics you've had, but it's really interesting to think about the social consequences of some of those metabolic processes you've been talking about. And I'm excited to see what comes out of your research in the coming years. So I'm sure you've given our people something to think about as well. So thanks for that. I'll say goodbye now, and thanks everybody for attending. And we'll have another presentation in a couple of weeks. I can't remember who it is this time, but Aldina will be able to tell us when he gets back. So it's good to see you all, and thanks again, Jordan. Thanks a lot.
See you later. Thank you.
Thank you so much. Bye.